First of all, good evening, everyone. On behalf of Biopractify, Biolab's research division, and our collaborating partners, I warmly welcome you all to the session 77th of our speaker series, A Beyond Research, a series where curiosity meets conversation. And today's session is titled From Where It Started to Where It Is Going, a research journey across borders. We are delighted to have with us Miss Palak Varma from School of Science, RMIT University, Australia. And we are really delighted to welcome Miss Palak to this session. And before starting, I would just like to share a brief insight into her journey. How Palak's journey began in India with a curiosity for science that gradually developed into a strong passion for research and innovation. She completed her master's in biotechnology at RMIT University, where she gained hands on laboratory analytical experience through academic and practical projects. And moving to a new country brought both opportunities and challenges, helping shape her resilience, adaptability, and professional growth alongside her academics. She also handled leadership responsibilities in the hospitality industry while continuing to grow in research. She has now begun her PhD journey, marking another important step towards contributing to a meaningful scientific research. And today she'll be sharing her experiences, learnings, and insights on navigating research opportunities across borders. And without further delay, and I would like to now invite Ms. Palak to take over the session. Over to you, Palak.
Thank you, Vineet, for the introduction. So, before I start, I'll just start with sharing my screen. I hope my screen is visible to all.
Yes, it is.
Yes. Okay. So hello everyone, and before I start this presentation, I have a question for all of you. Okay, so how many of you have at some point in your life searched things like how do I become a scientist or how do I get into research? If you have, please raise your hand. Anyone? Yes, I can see some hands. Yes, good. Okay, I did at some point too as well. So, good, good. I can see some hands. So, what do you think research is about? Take a minute to think and feel free to share your thought. What does research mean to you? Like, is it working in a lab, using microscopes, or performing experiments? Anyone? Good answer. Innovation. Yes. Exploring new ideas. Yes, that's right. You're going on a right track. Yes. So for me, research was all about curiosity. It is like using the existing knowledge to generate something new, like new understandings, validating hypothesis. Yes, that's that is correct, but that is a part of research which you do it. So let's take it forward. So I'll begin by talking about my journey from India to Melbourne, and I would really like this to be more of a conversation. So feel free to engage as we go rather than just listening. It's better you share your thoughts as well. So I still remember like back in 2020 when I was at home and like many Indian students who choose the science stream, I was preparing for NEET. And I'm sure like most people who are from India have heard about it. The entrance exams for medical colleges. When you take biology in school, the expected path is pretty clear. you prepare for NEET, you get into medical college and become a doctor. So, how many of you have felt the same pressure? Like, there's just one fixed path that everyone expects you to follow. Does the CGPA matter? My CGPA is just 8.5 and I'm currently doing my RP final year. Hmm. Hi Pratima. So we'll get back to the question. Like after I finish my presentation, it would be great if we gather the question at the end. So how many of you have felt the pressure like there's just one fixed path that everyone expects you to follow after your 12th standard? I can see some hands. Yes. So that was me. Like I was preparing for NEET and then suddenly COVID happened. And here's the best thing that this lockdown gave us. It gave us a real time, like to pause and think. So I found myself going down this rabbit hole and I started watching lectures on cancer biology, molecular biology on many platforms like Neptel, MIT Open Course, Coursera, just out of curiosity. No exams, no marks, and just having those questions like, why does this happen? How does this work? And that's really where my curiosity started. So I had this curiosity about cancer, but the next question was, what do I actually do with it? Let me ask all of you something. If you really wanted an answer to something, but you had no experience, no one to guide you, what would you all do? Anyone?
Ma'am, can you repeat it?
Sure, sure. So, if you wanted an answer to something, but you had no experience and no one to guide you, like what would you do? Find answer to it based out of curiosity. Try meeting industrial professionals. Yes. Search Google. Yes. That's correct. You're going on a right track. Yeah. So, yeah, refer research papers. Yes. So here's what I did. I knew I was curious, but I also knew that curiosity alone was not enough. I needed exposure to real research. So I started reaching out to professors and try to imagine during 2020s there was no chat gpt, no Gemini, no tools to make your writing sound smarter. So I had to read through the research papers, trying to understand their work, writing emails to supervisor I have never met, and explaining them in like just two paragraphs that why should they give me a chance? A student with no lab experience, no publications, and honestly, no idea about how really the research works. So I sent emails. There was silence, no reply, nothing. I kept checking my inbox again and again, hoping that something would change, but did not. Then I took a step further, which is a bit intimidating. I started calling them. At the below of their website, you have their email address and telephone number. So I kept calling them and I kept reminding myself that the worst they can say is no, but I had to try. And eventually, someone said yes. So I got an interview at ACTRAC, Advanced Center for Treatment Research and Education in Cancer, one of the India's leading cancer research institute in Mumbai. And that is where my research journey really began. Another important thing I learned along the way, and I want to say you that research is not just about working inside the lab. It is also about putting yourself out there, attending events, conferences, seminars, volunteering, meeting people, building a network that helps you grow both personally and professionally. So now I'm going to take a pause here and explain what I was actually studying, because I want you all to understand what the real world look like. So some of you might be familiar with these terms and some might not. So let's build this together from the basics. And if you have any doubts, just feel free to drop in the chat box. So imagine your DNA is like a book, okay? And every single cell in your body has exact the same book, our same DNA. But here's a question for all of you. If every cell has the same DNA, then why is a liver cell completely different from a skin cell? Anyone?
It's due to differentiation.
Differentiation, not completely correct.
Maybe due to different gene expression.
Yes, yeah, the right track. You're going on the right track. Yes, it has been specialized for that function, Janvi is saying. Yes, but how do you know that liver cell has a special function which skin cell doesn't has? The answer is something related to gene expression. So, in simple terms, it is about which pages of the book the cell is reading and how loudly it is reading them. So, my liver cells, imagine, would read pages from 1 to 3, whereas my skin cell would not read those 1 to 3 pages. It will read page from 4 to 6 and vice versa. So, some pages are active and some pages are silent. Now, let me ask you this. What do you think if a cell starts reading the wrong page at wrong time? So if your skin cell is reading the pages of your liver, or if your liver is reading the skin pages, what will happen? Mutation? Not completely right, but going on a good path. Anyone else? Okay. So that's actually what happens in cancer. Yes. So the cancer cell starts reading the wrong pages at wrong time and at full volume. Yes, proliferation. Yes. Everything becomes unregulated. What I was studying was something called chromatin modifiers. So think of your chromatin as the packaging around your DNA, and it controls which parts of the book are open and which are closed. And chromatin modifiers are like editors. Some of them add marks and some remove them. Based on that, they decide which part of the DNA should be accessible and which should be get hidden. So here's an interesting question for all of you. If you were trying to understand why some cancer cells survive treatment, what would you look at? Try to think like a researcher here. Euchromatin, heterochromatin, yes. Evolution, okay, that's okay. So let me show you how I approached. So I focused on the cancer cells that survived treatment because you all must know that not all cancer cells die. Some adapt and some continue to grow. So, what we call them is drug tolerant persister cells, which become tolerant to drugs, just like a bacteria becomes resistant to a drug. So, I asked a few questions. Which proteins do these cells have more of? Which ones are reduced? Where exactly the proteins are located inside the cell, whether it's inside the nucleus or in the cytoplasm or it's in the extracellular matrix. And are the chemical modification different of these cells with the normal healthy cells? So, in simple term, what I was doing that I was trying to understand how cancer cells rewrite the rules of system to keep them surviving. And once you understand that, you are one step closer to figuring out how to stop them. Again, let me ask you something. How many of you have thought about studying abroad and felt completely overwhelmed by the process? Yes, I can see some hands. Yes, yes. It's exciting, but also a lot to figure out. Yeah. So, coming to the next stage after my bachelor's, that's when I decided I wanted to go abroad for my master's. I did what I think almost every Indian student does. My dad and I made a spreadsheet. Countries, university, tuition fees, visa rules, post study work rights, English requirements, Australia, Canada, Germany, USA, and what all you can't think of. Everything mapped out. It looked very organized, but honestly, it also made things feel like very overwhelming. So, I want to ask you something. When you think of applying abroad, what do you think matters the most? Anyone? SOP? Yes. Research project SOP? Yes. Cost? Research experience? Good. Lab environment? Mhm. Internships? Proper guide? Correct. Yep. Those are great points. Most people focus on exactly these things. But I would say if you want to be a researcher and if you want to keep your research path open outside of your country, what I did was most people usually skip it. I started emailing professors before I even had an offer letter. Now, think about that for a second. Reaching out to professors when you're not even officially in the system. I would read their research papers, try to learn about their lab, understand their work, and find something very specific to ask, not a generic message that I want to learn this from you. Instead, something like, I read your research paper on this topic, and I was curious about this specific part. Do you think it could apply to this situation? Because here's the key, when you're applying outside, it's not about you, it's about showing that you have done the work, you have taken the time to understand what they do, and you're genuinely curious to get to work with them. Let me ask you this. If you were a professor, who would you reply? Someone sending the same messages to 50 people, or someone who clearly understands your work and asks a thoughtful question? Clearly understand the work? Yes. Work. Yes. Work. Exactly. That's the difference. That small shift changes everything. You move from just another applicant to someone they actually remember. And that's how things started working for me. That approach, that effort, those conversation eventually led me to some interviews and opportunities in Melbourne, Australia. And looking back, it wasn't just about getting in, It was about learning how to approach opportunities differently, not waiting for them to come to you, but going out and creating them. So, here's the story that gets really interesting. I came to RMIT University thinking I would continue in the same space I already worked: cancer biology, therapeutics, cell biology, molecular biology. It became like a comfort zone to me. But my master's project turned out to be completely different. It was computational work. Had to perform molecular simulations, perform docking, generate models, in silico research. Very different from what I had to do at Attract. And then my supervisor introduced me to Vipul Bansal. Let me ask you one more thing. Has anyone ever had a conversation that completely changed your direction? Like just one meeting that made you rethink your entire plan? No? 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 Okay, it doesn't happen often, but when it does, it really stays with you. Because I had a plan that I'll work into cancer, I'll work for therapeutics, but that moment was the completely different moment for me. Professor Bansal works in nanotechnology and diagnostics, a completely different field from where I started. And he introduced me to a project that I had never even thought before: building a rapid diagnostic device for Group B streptococcus. Now, think about that shift for a second, from studying cancer biology to working on diagnostics, from understanding disease to trying to detect it. And that experience taught me something very important. Your curiosity should not be limited. Let me ask you all this. How many of you feel like you need to decide one field and stick it to forever? Yes. Yes. Mm, no. That's a very common feeling. You feel like you don't have to move out of your comfort zone. And on the other hand, a lot of students feel that pressure. But I would say biotechnology is very huge. You have medical, food, environmental, marine, industrial, bioinformatics, and each of this has so many sub areas. It's easy to feel confused. So, here's what I learned: don't restrict your curiosity to one label, but at the same time, don't try to chase everything. Explore broadly, but focus deeply. The balance is what helps you grow, because sometimes the biggest opportunities don't come from staying in your comfort zones, they come from being open completely to new directions. So try to explore other fields, other areas where you can develop yourself. Now, let me ask you something. Has anyone here heard of GBS, Group B streptococcus? Yes, yes, yes. So, some of you are familiar with it. So, GBS is a bacterial infection that affects about like 1 in 4 pregnant women. And here's the concerning part: the baby can pick it up during the birth, and it can lead to serious conditions like meningitis, sepsis, and in some cases, even death in newborns. So, now think about this. Most women don't even know they have it. There's no pain, no obvious symptoms. And let me ask you this, if something this serious had no symptoms, would you even think to get it tested? No, right? Yes, that's the problem. Most people wouldn't. And even when the testing is done, current methods can take days to give result. So the goal of our project was simple to say, but it was difficult to achieve. They had to build a device that can detect GBS in minutes. At clinic, no lab required. One quick question. How many of you have taken a COVID rapid testing? Yes, Pratima, rapid testing. Yes. Yes. You all have already used something very similar. So this test is called a lateral flow device. And let me explain you simply how it works. So there are antibodies that are present and recognize very specific target. You can think of them like a lock that only opens with a specific key. In our case, the key is the GBS bacteria. Now, here's what happens. When you add your sample, it flows along the strip, and if GBS is present, it gets captured by the antibody. And then you see the colored line appear. Color line appearing on test is positive and no, that is negative. Simple to use, but not simple to build. And this is where nanotechnology comes in. Let me ask you, what do you think that colored that creates the colored line you see here? Antibody, antigen, substrate mixed with nanoparticles coated with antibody. That's right, Pratima. Yes, it's something very tiny. It's the gold nanoparticles, tiny particles of gold that cluster together where the target is detected. It creates a signal which your eyes can see. So in a way, you're looking at in a rapid test is not just a line. You're looking a nanotechnology happening in action. It's the same basic principle as a COVID test, but in our case, it was designed to detect a bacteria. And that's what makes this work meaningful. Because sometimes the biggest problems are the ones people don't even realize they have it. So after my master's, I stayed because I wasn't done. I had a plan that I want to pursue PhD since I was in my bachelor's. So now I'm doing PhD in applied chemistry and my project has leveled up. I'm now building aptamer based lateral flow devices for STI detection, which is sexually transmitted infections. Now I know, like, you must have heard lot of new words. So let's break this down together. Aptamer, I know it's new words for you. So what do you think aptamers might be? Anyone? Single strand of DNA, RNA. Right. Interesting. Yes, that's where you're going, right? So, aptamers are sometimes called synthetic antibodies. They are very short strands of DNA or RNA that are engineered in the lab that bind specifically to your target. Now, think back and connect back to what we discussed earlier. In the lateral flow devices, antibody act like a lock that recognizes specific key. So here, the aptamers are doing the same job, except Instead of proteins, we are using nucleic acids that we design. Now, let me ask you one more thing. If antibodies are already working, why do you think we need something new to design? Why do we need to make something? Anyone?
They are not specific enough.
Antibodies are specific if you choose for monoclonal antibodies.
But they are not gene specific. They can target particular cells, but not like gene specific cells or something. My opinion.
Mm, That's not completely right, because since we are not targeting the gene is something which is like present inside your cell, inside your nucleus. And in that case, if you have to go, we have to break all the cell and lyse and then take out the material. So that would be difficult even for an aptamer or even for an antibody. So I'll tell you the answer. Because antibodies are made using animals. Like you inject your antigen in animal and then you take out the antibody from the spleen and then you grow via hybridoma
So
technology. Yes, so that process is slow, expensive, and sometimes variable. Aptamers, on the other hand, can be designed and manufactured in a more controlled and consistent way, and is very stable compared to antibodies and can be stored at room temperature. Whereas antibodies you need to store at minus 24 degree, minus 80. So it is very important for diagnostics. And if you want to create a diagnostic test, I want all of you to think that if you want to create a diagnostic test that works in a remote area, place, places with limited resources, limited storage, limited infrastructure, what would you need? You would need something stable, which is affordable and easy to use, right? Yes. So that's what my PhD work involves, where I'll be working on developing aptamers that can specifically recognize targets like sexually transmitted infection pathogens and integrating them into lateral flow device. In simple term, it's taking everything I've learned so far: diagnostics, nanotechnology, molecular recognition, and bringing it together to build something that can be actually used in the real world. And that's the goal. Not just understanding the problem, but detecting them, building solutions that are faster, accessible, and can reach the people who need them the most. So, what do you all think? What is your image of a PhD student? All time lab rat. Conducting their experiments, more friendly professors, finding a new something interesting. That's interesting. I'm hearing things which I haven't. Long working hours, ton of research workload. Good. Honestly, you're not entirely wrong. That's very much correct. But also post lab work life balance, yes. Coming to Australia has really surprised me. So when I started at RMIT University, one of the first thing I noticed was the culture. My supervisor is Vipul Bansal, and he's not just my supervisor, but a mentor who trusts you, challenges you, and helps you grow from day one. And that small thing changes a lot. You're treated like a junior colleague, not just a student, and your ideas are heard and opinions are mattered. Better understanding, having deep knowledge about their point. Yes, yes. So, how many of you think research is something you can do alone? Anyone? No, it's not possible. Yes, can't do it alone. Yes. I had that bus, it's fading rapidly now, not possible. Yes. In reality, it's highly collaborative. In my work, we have people working from chemistry, engineering, chemical backgrounds, all working together. All of them have different perspective, but we are working on the same goal. And another thing that really stood out to me was work-life balance. How many of you think that PhD means no life outside work? I think Noor has said that I think it can be done alone if we have the required resources like data and funding. I completely agree you can do it alone, but on the other hand, if you do it alone, it's a very tedious task. At some point in your life, you'll need someone who can help you with things, who can help you figure out, who can help you troubleshoot. You can't be you can't understand each and every perspective. At some point you need someone, like if you're stuck at a bioinformatics part, you'll need someone who knows data science, you'll need someone who can analyze your data. Not me, since I see my PhD seniors have their life at their pace and links. Will get better exposure on collaborative or ender. Not me, since I see my PhD. Yes. Yes. There's a structure, there's a life balance here. You're encouraged to grow, attend conferences, seminars, have a get together every month or two. You, you don't just work. And that completely changed how I see research. Something you build over time with skill, patience, and creativity. There is very high confirmation bias when doing it alone. Yes. You get better exposure on collaborative or interdisciplinary work. Yes, when you get exposure, yes, that's completely right. So let me ask you more questions. How many of you are now thinking about pursuing research? Krish says me. Who else wants to pursue research? I can see people raising their hands. Yeah, that's really great to see. Even if it's just a small thought right now, that's how it starts. But if you take away anything from my journey, it would be this. First, master your basics. Let me ask you this. What do you think matters more, big dreams or strong fundamentals?
Obviously, strong fundamentals.
Yes. Kalvi says
Strong
both.
fundamental requirement.
Yes, strong fundamentals. That's absolutely right. My interview at Attract did not test how ambitious I was. It tested how well I understood the basics. And you might not believe this, but I was asked very basic questions like what is the difference between positive and gram negative bacteria? And later I heard that even some master students struggled simply because their basics was not cleared. Because no matter how advanced your work gets, everything builds on the foundation. So it's a mandatory that you all should have your basics clear. Second, be curious because research is not always exciting. You'll see experiments fail, results don't make sense. Sometimes you get the result which you don't expect to see. Okay. So, what do you think keeps someone going in that situation when your results doesn't align with your hypothesis? Anyone?
Ma'am, your voice is scrapping. Can you repeat it?
Yes, yes. So, experiments fail, results don't make sense. So, sometimes you get the results which you don't expect to see. So, what do you think keeps someone going in that situation when your result doesn't match with your hypothesis? Ambition and belief in yourself. Yes.
I think, ma'am, first you have to know that what is the requirement of your research, like empathy of people's. Then you can design based on that thing, and now then we can move toward to innovations, I think.
Yeah, that's a part of it. But imagine if even after believing in yourself, you have instances where you break down, where you feel demotivated. How will you handle yourself? What will be the one thing which will tell you to keep on going? Mental. That's good.
Ma'am, failure is a part of research innovation. So I think it not to uh, take a pressure about it because it is a part of innovation.
I understand that it's a part of innovation, but PhD for 3 or 4 year, it's a commitment, right? So what will be the one thing which will be telling you to okay, even if it's failing, even if you're demotivated, what will push you? Strong reason. Yeah. So I'll tell my personal view. So I think that curiosity is what pushes you to ask why something failed, what you can learn from it. I still remember my supervisor telling me that everyone celebrates the good results. Oh, you got it good. That's a good thing. But it's the unexpected result that teach you the most and push you forward. So I would suggest every time be curious, even if it's good, even if it's bad. Be curious and let that thing push you forward. Third is reach out early. Don't wait for the perfect time. Let me ask you one thing. If you had to email your professor today, what would you say? Anyone?
So I recently got into IISc internship. So what I did was my perspective, like I genuinely, like you said, I went through some of his research papers in Google Scholar and other medium, and then
Yes.
I saw how it is related to my current interest, or how it is aligned to my current interest or research work, and I genuinely like told how interested I am in it, and how my ideas, I mean, his ideas are so aligning with
Align.
mine, and
Yes.
yeah, and how I can get to know more about it. And then really, I got into it actually. So I've also mentioned not just my current college projects or undergrad projects. I've done some science projects related to it in school too. I've attended plenty expos and stuff. So I mentioned those
Mm-hmm.
projects too. So that way, I got into it.
Yes. That's absolutely the one thing. That's very good you did. And congratulations. So here's
Thank
also
you.
what yeah, here's also what actually worked for me. Read the last two, three papers, find something that genuinely interests you, and ask one thoughtful, specific question. Because a thoughtful email stands out more than a generic message, more than just a CV. And that's really been my journey. Not a straight line, but a series of small steps, questions, moment of curiosity. And if there's one thing I hope you take from this, it's that you don't need to have everything figured out. You just need to start and things will fall off. One last thought. So for me, research is not a straight line. It's hundreds and millions of small choices to stay curious and one quiet decision to keep going. Thank you, all of you, for joining this session and making it more interactive and sharing your views. And if this resonated with you, feel free to connect with me on LinkedIn. I also share my journey and insights through my newsletter, and I would genuinely love to hear what your thoughts are. Thank you, Vinith, for organizing this and giving me a chance to be a speaker on this platform. Thank you.
Thank you very much, Palak, for this insightful session. And it is really surprisingly, interactive. And we have never seen, such interactive sessions for a, speaker. So it is really good. And, I would
Thank
just
you.
like to, have a vote of thanks presented to you by one of my team members. She'll be, presenting the vote of thanks for you. So, Ayushi,
Hello, ma'am. Thank you
Hi,
so much.
hi.
And thank you so much for such an insightful and inspiring session. It was truly wonderful to hear about your journey from India to pursuing research opportunities. Sorry, for opportunities, along with the experience and lesson you shared throughout the sessions. The session turned out surprisingly interactive and engaging, with participants actively asking and connecting deeply with your experience and insights. It was filled with a valuable guidance for our students and inspiring researchers who wish to explore opportunities in science and research across borders. We sincerely appreciate you taking out your valuable time to connect with our audience
Thank you.
and share your experience with such honesty and I would like to also thank to all participants for joining us today and making the session interactive through your questions and participations. On behalf of Biopractify and our collaboration partners, thank you everyone for being a part of today's session. We hope to see you all again in our upcoming sessions beyond research where curiosity meets conversations. Thank
Thank you
you.
so
Have a lovely day.
much.
Thank
Thank you
you.
so much for joining, guys. Thank you.
yeah, and also, if you can share, the link of your newsletter, I would be really it would be really great for the participants, as I have seen it. It is really insightful. So
Sure.
Maybe if not, if it is available now, then now, or maybe we can also send them to through the community.
I can share it now as well, and if you want, I
Yeah, that would
can.
be great then.
Sure, I'll do it. Yes.
So once again, thank you very much, Palak. And it is also really great to see your journey from, when I think initially when we connected through you being a participant in one of our skill development
Yes.
programs. So from there, like I've been, sort of aware of your journey that you have been traversing. So it is really great to see you grow in your role and achieve different things, and
Yes.
really happy to see you growing this way. Thank
Thank you so much, Vinit. Thank you.
You're welcome.
Thank
And we would also be happy to host you for more sessions. Like I think because the way questions are, I think there are still some questions left, so we might need you for another session.
Sure, that definitely works. We can do another session. Yes.
Great. Then, without thank you everyone for joining. Then we may conclude the session now.
Thank you guys. Thank you for joining. Hope to see you next soon.
Yeah.
Have a good night. Bye bye.
Bye bye, everyone.